We will now explore how the genetic code in DNA is used to determine our characteristics. Our characteristics like hair color, eye color, and blood group are determined by proteins. For example, the pigments that make your hair colored are proteins. DNA carries the instructions to make these proteins. A section of DNA coding for a protein is called a gene. The DNA in our cells is in a special compartment called the nucleus. Another molecule called messenger RNA or mRNA carries the information in DNA to the rest of the cell. The mRNA carries the code for one gene and is used to make a protein. RNA or ribonucleic acid is similar to DNA. The nucleotides in RNA are slightly different in structure. RNA contains ribonucleotides. DNA nucleotides have one oxygen atom less and are called deoxyribonucleotides. RNA has only one strand, whereas DNA has two strands. We saw that DNA has 4 flavors of nucleotide bases: A, T, C, and G. RNA also contains A, C, and G, but instead of T, it has U. U is similar to T with a small variation in structure. So now U pairs with A. To make mRNA from DNA, we need access to the sequence of nucleotide bases. So the 2 strands of DNA are first pulled apart in the region of our gene. This is similar to DNA replication, but only one gene is involved. mRNA is synthesized by complementary base pairing between the DNA and RNA nucleotides. Since it carries the code for one gene, mRNA is shorter. It is single-stranded. This means that only one DNA strand is the template. The mRNA sequence is complementary to the template sequence. Since the second DNA strand is also complementary to the template strand, the mRNA sequence is identical to that of the second strand. This strand, which carries the actual code of the gene, is called the coding strand. This process of converting the code in DNA to the code in mRNA is called transcription. A DNA or RNA strand is always synthesized from its 5' end to its 3' end. The reactions only work when new nucleotides are added to the 3' end. The chemistry behind this is out of the scope of this course. Recall that the 2 complementary strands are antiparallel. If the RNA is synthesized from 5' to 3', the template is used from 3' to 5'. This means that the DNA sequence is always read in the same direction when RNA is made. Different genes have different coding and template strands. For another gene, the first strand may be coding and the second strand may be the template. For this gene, this is the coding strand and this is the template. The cell begins transcription only at specific sequences of DNA. Such a sequence is called a promoter. Promoters are found before the gene and indicate where the gene begins. They also specify which strand is the template. Promoters are not part of the code of the gene and only act as sites which allow transcription to begin. A gene may be 20,000 to 30,000 base pairs long. The promoter of the gene is shorter with 100 to 1,000 base pairs. You now understand transcription. In the next video, we will see how mRNA is used to make proteins.